So, first of all, I'd like to thank everyone for inviting me. I mean, you guys are doing a wonderful job at getting the information on PCD out there for everyone. And I mean, I'm guilty of not being able to be present at these meetings all the time. So it's great to be able to have a recording and dip in and out whenever you want to on the website. So long may it last, guys. You're doing a great job. So today I want to essentially just give you an overview of a little bit of history of diagnosing PCD using cell cultures, and then go on to some of the advances and some of the pitfalls that we fall into. I'm not going to take up an hour and a half of your time, so don't worry. So this is us. We are, just see if I can get this going next. So this is us. We're at the Centre for PCD Diagnosis and Research. We are an NHS highly specialised commissioned service, and in Leicester we serve the Midlands and the north of England. We're based at the Hodgkin building at the University of Leicester, and we serve a population of around about 30 million people in the north of England and the Midlands, and we take biopsies from multiple teaching hospitals, and they're couriered to our centre in Leicester, where we do all the diagnostic testing in the lab. And they're listed here. To date, we've tested over 3,800 patient biopsies in our clinic. Of those 3,800, we've diagnosed approximately 600 PCD cases that were previously undiagnosed. And pertinent to this study, I got the guys to look at how many alley cultures we've done, and we've done over 2,000 cultures. So, you know, we have been using this for a long time now. This picture here on the right shows 1 year's worth of our workload, essentially. So these are where the patients live that get biopsied and the samples sent to us. The green ones are the negatives and the red ones were the positive samples, which had PCD. So just to give you a bit of a background history of how I came about doing this. In 2005, I was a postdoc working with Prof. O'Callaghan looking at meningitis research. And he came to me and asked me to help develop the method for culturing the nasal brush biopsy progenitor cells. Now, I'd spent a time, I'd been over in Australia and worked on stem cell therapy for a couple of years. So I thought we might be able to help out here by using some of those techniques and applying them to get the cultures going. So the reason he wanted me to be able to do this was so that it would support and go into the application for the National Specialist Commissioning Group application to fund the PCD diagnosis across the whole of England. And the original application was done by Chris O'Callaghan, Andy Bush at the Royal Brompton, and Professor Warner in Southampton. I think John Warner moved then to Imperial. But essentially, we had a problem in that the amount of cells that we were getting back from a brush biopsy were tiny. They were invariably infected, and the cilia also had lots of kind of inflammatory damage or physical damage. So I looked at the literature and I've got two choices. I could either do the submerged technique that Mark Jorrison had pioneered in Leuven, or use the quite new at the time. I mean, okay, it was new to us, but Larry Ostrowski in Chapel Hill had been doing this since 1995 by growing the cells at an air liquid interface with a basolateral compartment where the nutrients are fed into the cells. So the spheroids, first of all, because Mark was very close, I thought, well, I'll just email him, and he very kindly accepted me to go and visit the lab. So I went over there and discussed the technique, what he was doing, and it was fantastic. They essentially used surgically excised turbinates, which are the kind of parts of the nasopharynx that can get inflamed. And then he took them out surgically. they digested the cells, and there was loads of them, and they grew them in kind of a jelly, collagen jelly. And then the cilia could be grown as spheroids, they called them. And essentially what they were little tiny bundles of cells with cilia on the outside that spun around in a circle. And I thought it was wonderful, but the problem that we had was we didn't have that many cells to start with. So in order for me to do that same technique, I would have needed a hell of a lot more tissue than a brush biopsy. So I decided that we should pursue the alley culture really and adapt Larry Ostrowski's technique. So what was known about the alley technique when I first started was that in 1993, Scott Randell's group at Chapel Hill developed the rat model. So they used Ham's F12 mix with collagen EGF, retinoic acid, and air exposure on these transwells in order to get this polarized airway liquid interface culture going. And it wasn't until 95 when Larry Ostrowski, again from Chapel Hill, honed it for humans. And this was in lung epithelium though. They hadn't actually grown nasal cells before. So it was a bit of a, can we do it for nasal epithelium from a small biopsy that only contains 5,000 cells? So the questions were, can we regenerate the complex epithelium? Can we enhance the PCD diagnosis? And can it be used as a research tool? So those three things were really the driver for what we were doing. Okay, I've lifted this from a fantastic review that I recommend that everyone reads. It's by Maria Legrande and Hannah and Laurie Emmanuel Zaragoza. So what they've shown here in this review is a transcriptomics of the alley cultures, and you can see that this layer of cells, the different colors, represent the different subtypes of cells in the epithelium. And the ones that we want to propagate are the basal cells here. These are cytokeratin 5 positive. And then there are cyclin basal cells, which fail to express MK167. And then you get the suprabasal cells, and you can see they're moving towards the ciliated apical surface. But essentially what you can do is look at the transcription factors in each cell subtype and then sort them so you can look at the population of cells over time on an alley culture. Now, from a brush biopsy, you also get lots of other contaminating cells like smooth muscle, endothelial cells as well. Okay, so this again was from the same review, but it's very up to date actually, and shows how we get from a basal progenitor cell through the different cell types to become a multiciliated cell. And what you find is that the signaling mechanism is notch. Now, for those that don't know about notch signaling, it means that notch is produced very close to another cell. Notch is degraded if the cells are too far apart. It's not a very stable compound. So essentially the cells have to be together so they've got the notch signaling working very well. So it's important that basal cells are cultivated very close together, and they essentially turn over. And then when we have very high levels of notch, this program kicks in where we get MCIDUS, which is multicillin, as one of the drivers, CCNO, also a driver for ciliogenesis, and also FOXJ1 for ciliogenesis. So we get this club cell, which is thought again, some people say that this can self-renew. Other people say that it is just a transitional state of a basal cell into a columnar epithelial cell. But what we get is the deuterazone protein starting to drive this amplification of the centrioles before we start to initiate ciliogenesis. So what's interesting actually about these proteins, the ones in red, the transcription factors and the proteins in red are that they've been shown to be causative of PCD or a PCD-like phenotype. So what is the method then? How do we actually get the biopsy to become ciliated? And this schema here, it's very busy, but I'll go through it carefully. We start with a brush biopsy, and then we incubate it overnight in primicin, which is a designed antibiotic, antifungal agent to keep primary cultures sterile. So the idea there is that if there is any contamination in the brush biopsy, we get rid of it overnight. And then what we do is we put the basal cells on a collagen-coated 12-well plate here, and then they grow to pretty much confluency. When they are confluent, we then do a trypsin EDTA treatment, break them apart, and then reseed them on a collagen-coated flask, a T75 flask. And they grow then to a density of about 3 to 5 million cells. And we use this media system called Pneumocult EX, which is the expansion media. And they're fed every 2 days, so the media is removed and then we put new media on top. So what happens when they're confluent in the flask is we do another trypsin EDTA digest, get the cells out, and then we seed the cells on 1.2 centimeter squared transwells, which again are collagen coated, but we use the expansion culture media, and then when they're confluent, we change the expansion media to the Pneumocult Ali formulation. So we plate around about 500,000, so 500,000 cells onto there, so they don't take long to be confluent, just a day or so, or even overnight. And that closeness of the cells really does help with the differentiation program and the ciliogenesis program. So essentially, this period, once you've got them on the transwell and they are exposed to air, you do have to wait a little bit for the cilia to start to form and to mature. And that can take anything from 3 to 6 weeks, depending on the patient donor, how many cells you started with, and whether or not there's any kind of issues with the with infections or anything else along the way. But on average, we were getting cilia formed around about 15 to 28 days. Okay, this is data from my lab where we looked at the ciliogenesis timing at different days. And you can see here that on the alley cultures, we don't see any cilia at all, but we use this centriolar stain called CNAP1. So CNAP1 is expressed in the centrioles, and you can see you get multiple basal body forming by day 12. We have 238 cells per well. So in 1.2 centimeters squared, there are still very few cells that are starting to initiate that multiple deuterosome centriolar formation. And then the green here is the acetylated tubulin, which is present inside the cilia. And you can see we have CNAP1 positive cells at day 17, but also acetylated tubulin positive. So the cilia are just starting to sprout from the cells at this time scale. And then by 21 days, we have full length cilia, which are about 5.6 microns in length, we think. Now, when I was doing this talk, I was really reassured by this paper by Pascal Barbary and Laurie Emmanuel Zaragothi. They're really pioneering the single cell sequence analysis for the transcriptomics and identifying these alley cultures in nasal cells. And this here shows the populations of cells in different colors and the time in an alley culture from a nasal brush biopsy, how they become ciliated towards the end. So by 28 days, you can see that a lot of the cells are club cells, goblet cells, but also you've got a huge portion of multiciliated cells at the end. So that was kind of reassuring when I read it, because the previous work wasn't published. So what we can do with the alley cultures is we can fix them and coat them to look at the surface characteristics. And this is very useful when we're looking at things like biofilm formation with bacterial infections, other things like mucus interactions with the epithelium. On the left, you can see a low power image showing the cilia sprouting from the cells. And on the right, you can see a high power scanning electron micrograph. So most of the alley cultures also produce apical fluid, which is basically ultrafiltrated media from the basolateral compartment being pushed up to the apical side. The oldest ciliated culture that we kept going in the lab was 11 months. So you can do long time courses with these cultures, which we found very useful. So I'll show you some movies of what they look like. This one, I don't think you'll see the cilia, but hopefully you'll see the particles moving on top of the cilia. So look out for, oops, sorry, let me just go back one. Look out for the particles moving on this. Can you see? There's particles moving over the surface of the cilia. That's 100 times magnification looking down upon the alley culture. So we can zoom in on that, obviously, and do it and record in slow motion to decipher the ciliary beat frequency and pattern. And here you can see under 1,000 times magnification, we're starting to see the cilia moving better. So what we can also do is lift the cells off the transwell, put them into suspension culture media, and then we can image the cilia inside profile. And that's what this movie is going to show you. You can see the cilia moving sideways. There's quite good clearness here, although this is in slow motion. And so we use this to kind of back up what we find in the biopsy when we do a diagnosis for PCD. Usually, yeah, it takes a little bit longer, but it's definitely worth it. Now, one of the things I wanted to show people who wanted to use this technique was, how do you know when you're getting really, really good cultures? Well, I want you to keep your eye here in this middle, and then there's cells all the way around it. Now, this is called a epithelial rosette, and you'll be able to see it when the movie plays. There's a good central cell, and then there's cells all around it. But you can see that the cilia are very widespread throughout the cultures as well. So you know you've got a good culture when you see the rosettes and you see lots of cilia. So in the early days then, what we wanted to do was to try and kind of look at more about the physical characteristics of these cells. And the TIA stands for trans epithelial electrical resistance measurements. And this is a picture of the chopstick electrodes here, where you have a negative and positive charge above and below the transwell. And what you're measuring is the electrical resistance in ohms across the cellular layer. So the thicker the layer, the more the resistance. That's essentially what it means. And when we looked at our cultures, it was quite striking for me how there was a huge difference in the electrical resistance. Now, does this relate to the apical flow that I was talking about earlier, that you may have lower electrical resistance if you've got a higher basolateral to apical flow rate? But you can see that all of these cultures were very well ciliated and were means of 4 readings per culture. So I worried really using this too much that you end up getting variable readings anyway from patient to patient. Obviously within cultures it can be quite useful, but across different patients it can be a bit misleading. And also what we found was when we were routinely using it, it does actually expose the cells to the risk of culture, let's say, from the chopstick electrodes. So the cultures, when we looked at the cilia speed, we can see here, these are 9 cultures that we did. These are the speed of the cilia in the biopsy. And then after culture, they were pretty much consistent. The PCD patients as well were pretty consistent in that the cilia were very slow or static. What we did see was that the speed of the cilia from the biopsy seemed to be faster than the alley culture. And whether that's because of the mucus loading on top of the cilia, we don't know, we're not sure, but that we always seem to get slightly lower beat frequency reading in the alley cultures compared to the biopsy. The other thing that we started to notice early on was that in a biopsy, you still get residual twitching of cilia in, for example, an outer dining arm defect where you don't get any DNAH5 expression. The motility still twitches from time to time, but when you culture it, they're almost static. So it almost kind of makes the phenotype of PCD more severe almost. But the fact is that they're abnormal and it was useful. So when we look at the ciliary beat pattern in the cultures compared to the biopsies, again, when we get a really healthy normal, there really was no difference in the dyskinesia scoring of the cilia. So the beat patterns were consistent before and after. But when we started looking at the ones that were really badly damaged from the biopsy, so these are the ones that have probably 50% of the cilia that don't look normal, but the other 50% are okay. We started to see after culture that the dyskinesia scoring went really down. So we thought, oh, this could be very useful for some of the cultures where they're either insufficient cilia to study or they're damaged. So we also looked at the beat pattern of the cilia from pre and post culture from PCD patients, and here you can see a range of different PCD types showing that they're abnormal on both before and after culture. So when we pulled the data, it was a bit of a surprise to me, but it was also very good to know that when we get really poor quality ciliated epithelium from a biopsy, we can culture them and significantly improve the actual beat pattern analysis. So what that does is it means that we don't have to repeat biopsy the patients. So it saves the clinician's time, it saves our time in the lab. And so it was an invaluable thing to have to be able to exploit. When we looked at the transmission EM of the normals, and this one's a static dining arm defect, you can see before and after culture, the ultrastructure is pretty much identical. We couldn't see any differences at all. So, I mean, when you present these kind of studies, it's very easy to fall into the trap of, oh, well, Rob does it and it's dead easy and blah, blah, blah. It isn't, it's really tricky. And along the way, we had lots of problems. And I'll just highlight some of these because if anyone wants to take this method on, you know, keep an eye open for these things because it can save you a lot of time. One of the things that we really struggle with is having very, very small numbers of cells to start with. Now that's down to the biopsy taker. If the biopsy taker isn't spending enough time in the patient's nose to harvest enough tissue, it makes growing the cells very, very difficult. The basal cells, you can grow them, but they become exhausted and the ciliogenesis program doesn't switch on. So the other thing that affects the success rate is the disease. So if the patient has an upper respiratory tract infection within the last, say, 4 to 6 weeks, we've shown that you cannot grow them. They just die in the culture tray. Now that might be because of viral loading or any other disease reasons. So the primicin isn't that good at getting rid of viral infections. So the other thing that we noticed early on was we had a problem with dehydration where the cultures was cracking around the edge. And that's where the mucus dehydrates and sets solid on top of the apical surface. And the cilia are beaten underneath. and the shearing forces will just pull the cells away so that the culture just fails then. So to get around that, you must increase the incubator humidity. Now we do that by putting a reservoir on each shelf where the cells are grown. So I touched on infections earlier, but what we can see is that, you know, viral infections are, and obviously with COVID, we weren't sure whether we were propagating COVID in our cultures. Now, what we did was we had a very strict regime where we tested individual patients who we biopsied to make sure they weren't COVID positive. But we did actually accidentally in the early days culture one that was actually infected with COVID, but it still the cilia still grew. So it was amazing how you know, the coronavirus doesn't actually completely inhibit the ciliogenesis program. The other thing we found was that if we have too many operators in the cell culture room, sharing each other's feeding and things like that, and splitting each other's cells, we quite often got infections. And that meant that, you know, because it's such a long investment of time, to get an infection in these cultures means that it's a real problem. So we limited to 1 or 2 operators at the max per culture. The other thing we found a problem with was sporulating both fungi and bacteria, where because we're exposed to an air interface, when you do normal traditional cell culture, it's submerged, spores can't really invade your cultures as easily. But because you're growing at an air interface, the spores can actually quite easily land on the cells and cause damage and infection. The other thing we had in the early days was we used this media called BEGM, which just wasn't working anymore for some reason. So whether it was a bad batch, a bad lot, we weren't sure. We also had a bad batch of collagen. So suddenly all the cells weren't sticking to the wells anymore, which made it like, what's going on here? You know, and it was down to the fact that the supplier's collagen was a bad lot, essentially. And they hadn't obviously tested it on all cell types. So overall, though, the lower CBF after the biopsy in the cultures is something to be aware of. And also the phenotype exaggeration of PCD in the cultures compared to the biopsy is another thing that you have to be mindful of. So we've got 3 centres. I mean, the guys here, Yinting, Katie, from the other sister centres, and they essentially use the same methodology as us in Leicester. In 2006, we got it up and running here in Leicester, and then I shared the protocol with the Royal Brompton Hospital and Southampton in 2006 and 2007. And then in 2018, we decided to review everything because we were having all these problems with BEGM not working, with collagen problems. So we decided then to look at the pneumocult alley medias instead. And this paper was driven by the Southampton team, and Janice Coles is the first author on that, and she and Claire Jackson really established this new method, really using different medias to improve the amount of cilia we get from about 54% up to about 80%. So instead of 5 out of 10 of our biopsies becoming ciliated, now we've suddenly got 8 out of 10 becoming ciliated, which was a huge improvement actually. And we compared each other's data sets. What Janice's paper also describes is a method for measuring ciliary frequency in fields of cilia using a fast Fourier transformation methodology, and also how to preserve the biopsies using cryopreservation methods. I'm not going to go into that, but needless to say, if you need any info, the Southampton team are great for sharing their expertise. Now, one of the things that came out of the paper was that Janice had to actually say that, you know, this is a complex specialized technique. We do actually get limited passage numbers out of the primary cells, which is a shame really, because if you could continually grow them, it would be brilliant for studies of comparison with infections and other things. So the other big problem with alley cultures is there's no glands, so there's nowhere for the mucus to drain to or the apical fluid. So, you know, we have to be also mindful that in vivo and in vitro, it's completely different. So there are always limitations to these models. One of the other things that I noticed was that we start to get these the ciliated cells lining up in a spiral. So you get these spiral formations where the cilia align themselves in a planar cell orientation, and it sweeps the mucus in like these whirlwind type things, these like whirlpools. And again, whether that happens in vivo, I'm not sure, but I doubt it. So the big issue, again, with the failed BEGM media and everything else is because we don't know, the suppliers will not tell us what's inside the media. So that's an ongoing issue. So this is data from our center alone, where we have quite a lot of new doctors taking biopsies who take really rubbish samples. And they essentially are insufficient. We call them insufficient or whatever. And there's no cilia in them to study from the biopsy. So what we do is we put the whole biopsy to culture, and we call it orthoculture. So what we did for BEGM in the early days was we put orthoculture anything that didn't have any cilia in it, essentially, but had a few cells. And we managed to get about 30% of the orthoculture cells ciliated, and then we managed to get the full repertoire of testing from the culture. However, when we switched to pneumocult, now we're getting 6 out of 10 of these cultures ciliated. Now, this is invaluable for the doctors because it means that they don't have to see the patient. If it's a barn door normal, it will be normal. And the clinical history, we've got the NO as well. So therefore, you can rule out PCD based on the ability to culture them. And if they're normal, they're likely to be normal. So when we looked at the PCD positive success rate down here, you can see with BEGM, we used to get about 56% positive cultures with static cilia or abnormal cilia from biopsies that were PCD positive. And again, that's gone up to now 82% thanks to the pneumocult alley. So it really is a fantastic media to use, although we don't know what's in it. So I'll move on now to how loads of people, us as well, have tried to make improvements to the alley culture because it's still got its limitations. So some of the things that people wanted to do, what I wanted to do early on, was really to get the basal cell proliferation rate up and the ciliation amount increased, and also the ciliated cell number to increase. So more of the cells on the culture becoming ciliated. So this is data from a paper that we published a while back where we used Rho kinase inhibitors. Now, Rho kinase is one of these small molecules which control the cell cycle. And if you inhibit the Rho kinase, you stop the cell from going into its quiescent mode. So essentially, it's in GS all the time. So it's in the growth phase all the time. And then when we looked at this Y here is the compound, which is the Rho kinase inhibitor. You can see with BEGM that the cell doubling times plateaued. Whereas when you put the Rho kinase inhibitor in, they continue to grow. So essentially, we're getting more proliferative capacity using a Rho kinase inhibitor. So in addition to Rho kinase, we used a feeder cell layer here. And our feeder cell, essentially what the idea there is that instead of collagen 1, you're giving an extracellular matrix for the basal cells to sit on top of, to grow and to differentiate into more pre-progenitor-like cell types. And we use Swiss 3T3 cells, which are fibroblasts, and they're mitogenically inhibited using mitomycin C. So the only cells that are dividing are the basal cells. And what we showed was that the technique of feeder cells and rho kinase inhibition, you can get massive amounts of cells back, huge numbers. And Sam James's team at UCL are trying to use this kind of technique to get enough cells to use for transplantation and cell therapy in respiratory diseases. So we also, I've got a bit of a bugbear with it, because obviously having the presence of another cell type, which is alien to the basal cell, the epithelial cell, you are getting genetic and proteomic contamination. So it limits what you can do with the cells a little bit. So this is data from my lab, where I had a master's student who looked at notch inhibitors, and this is a compound called DAPT, D-A-P-T. It knocks out the notch signaling, and if we introduce it at a time when the cells are starting to differentiate, you can get the ciliation program accelerated. So we went from 28 days in the control to the DAPT treated to 24. This culture was down to 19 days, and then this one was down to 17 days. So it actually, the cultures look more ciliated. There's far more cilia. They are, they secrete less mucus because there's more cilia compared to mucus cells. But one of the things that I noticed was that when we kept them going, they didn't survive for very long. So before we can keep the cultures going for 11 months, these guys died after about a month in culture. So I think the notch inhibitor actually makes the cells live fast and die young. That's what I call it. So, okay, so the other big thing that we want to do, obviously, is to treat PCD and get rid of it altogether. And in order to do that, we need to miniaturize these cultures so we can use high throughput screening, looking at robots, testing libraries of compounds, looking for any kind of way that we can make the cilia work better in PCD patients. So it would be useful also to look at other diseases, not just PCD, to get this method. So we managed to grow cilia, the alley cultures, in these tiny little transwells in a 96 well format, which allowed them to be put into these robotic high-speed pipetters. So this paper here that we published in 21 was showing that we could essentially use read-through agents which cause the ribosome to skip a stop codon. So if we've got a premature stop codon in a cilia gene, then the PCD's there. But if we could skip that stop codon, it would make potentially a full protein and it might be functional. So we added atalorin to an MCIDus patient who had a stop codon prematurely cited in the gene. And then what we showed was that we could get multiple basal bodies in the atalorin treated group and the gentamicin treated group, but actually we still couldn't get the cilia to grow. So whilst it helped a little bit, the read-through for PCD, there's still a long way to go. I think that Hemant Omron is in a position where he's looking at CCDC 39 and 40, and I think they've had a bit more success in getting normal cilia back. But one of the things that this high throughput screening is going to be essential for is for gene replacement therapy using CRISPR-Cas9 techniques to see if there's a proof of principle for repairing the cilia. So the other thing that we obviously wanted to do was to immortalize the basal cells so we can use them as a research tool in the lab. And there are a number of ways you can do this, essentially. There's a virus called SV40, which is a simian virus. It's a large T cell antigen, and it causes like a cancer type phenotype where the cells lose control of their cell division and keep dividing. And that works a treat for basal cells. So the problem comes when you want to differentiate them, because you don't get polarity and they become poorly ciliated. You get cilia growing down instead of up. It's just a non-starter. There's another viral vector called lentivirus, and I think that the Belgian team, Mark Jorissen's group, and Massimo Pifferi's group in Italy, they've had some good success with this, but we've not tried that. But what we did was we looked at this protein called BMI1, which again, it kind of locks the cells into this proliferative phenotype in the cell cycle. And we showed that we can passage them when they're BMI1 transfected up to about 23 times. Now, that's great that we can see here on this graph that the number of cells, even out to day 20, passage 23, so that's trypsinizing them 23 times, we can still get good doublings of the cells. The problem was when we start to differentiate the cilia from these cells in the later passages, the movement of the cilia was abnormal. So we're cautious about using them as a kind of a tool to kind of test some of the PCD agents. Okay, so there are other methods out there, and I'm going to just brush over this really, because I mean, we've done this in our lab for an experiment where I put essentially the basal cells on Matrigel, which is like a jelly, and you can have different concentrations so that the cells actually grow as like a lumen inside, and you get the three-dimensional structure forming, and that's what this is here. And what tends to happen is the cilia are usually on the inside, which means that they're really hard to kind of look at because you can't image the cilia in the lumen very easily. And also you get lots of mucus inside the lumen. So it is also technically difficult to do. But hot off the press, this paper was published in 22, so in August this year. The Wren group in Pittsburgh in the USA have got a method of putting the basal cells into a suspension culture first and then into Matrigel. And they managed to kind of turn the organoids inside out, essentially. So that the cilia, in red here, are on the outside, and they rotate. And they've looked at PCD patients, and they can recapitulate PCD patients using this too. So this might be a better way of doing it. But when I was reading it, I was thinking, hold on, this is like Mark Jorissen was doing in 1989. It's exactly the same pretty much, but you know, it is using slightly more knowledge that we've accumulated over the years, essentially, to kind of flip up the polarity of the basal cell from inside to outside of the organoid. So not only PCD, these cells can be used to study other things, and this is a quick overview of where we've used them in different papers over the years. We've managed to use to look at the early cultures from transplanted cells, from lung transplants, working with the Papworth team. We also have looked at obviously asthma and mechanisms of asthma in early cultures. We can look at hypoxia and cystic fibrosis using the early cultures. COPD, we've just had a paper published where we looked at that. We can look at the mechanisms of the apical secretion. So we think that aquaporin is very important in getting that transfer of basolateral to apical fluid flow. And then also obviously infection. Everyone wants to look at infection because pneumonia is a real killer. And this is a great method to use to start to understand and decipher some of those pathogenic mechanisms of action. So Ali really remains my preferred ex vivo and in vitro model over these years, looking at all the other different techniques, because you have so much more access to the epithelium to add things to. And so far, I did a quick search and I could find over 1,000 papers on PubMed. So in conclusion then, to finish off, we have shown that we can successfully ciliate alley cultures from biopsies. We can expand the basal cells. We can also freeze them now, cryopreserve them, and we can passage them and immortalize them. We can also harvest the mucus and apical fluid to look at some of the diseases such as mucus plug-in, COPD, all these kind of diseases that may involve the apical fluid and the mucus secretion. We can actually recapitulate the ciliary beat frequency and pattern and the actual ultrastructure before and after culture, which reduces the number of repeat biopsies we have to do on the patients, which is excellent, really, really useful. Now, we've also looked at some of the apical fluid, and we're looking at cytokines and nitric oxide released from the epithelium to look for inflammatory mediators. We can study ciliogenesis and obviously other epithelial proteins that may be relevant to other diseases and not only just cilia. So, we deploy immunohistochemistry and super resolution microscopy to look at some of those mechanisms and proteins that may be important. And obviously the alley culture is a really valuable source of DNA and RNA for looking at sequence analysis for genes and diagnosis, and also for therapeutics as well. Now, one of the areas that I think that we're lacking in the literature is understanding the post-translational effects of some of the proteins important for PCD. It's great showing that we have a variant or we have a pathogenic mutation, but what we don't know is what happens to that protein as it goes into the cilia. Does it actually get trafficked differently inside the axoneme? Are there compensatory mechanisms inside the protein architecture of the cilia that may actually help with the PCD patient? So all these methods really rely on a proteomic approach, looking at the post-translational products of the proteins when they're there inside the cell. And I think that is where the future of PCD is going. I can't do this on my own unless they're just one of three brilliant teams. These other two teams are excellent, and we will be much worse off without these guys. But essentially, Gwyneth in my lab does most of the cultures, and obviously Janice does quite a lot of the cultures at the Brompton, and I think Paul now is really leading everything at the Royal Brompton. So with that, I will stop and take any questions. Thanks a lot.
Thank you very much, Rob, for this very interesting kind of tour or short history tour of cell cultures in PCD. Let's kick off the Q&A. And first of all, we have a question from Charlotte. She asks, how much does only ROCK inhibitor treatment improve the basal cell proliferation without the feeder cell layer?
What was that question? Sorry, I didn't get the...
How much does only ROCK inhibitor treatment improve the basal cell proliferation without the feeder cell layer?
Now, it does increase the proliferation, the ROCK inhibitor on its own. The feeder cell layer really is there to expand the cells into a huge number. So yes, you can use ROCK inhibitors alone and they're fantastic. Hope that answers your question.
Yes, she says thank you. Excellent. And shall we have Mathieu next? Hi, Mathieu.
Hi, Mathieu.
Hi. Thanks, Rob. It's a great talk. Honestly, I could ask you like 10 questions because it's really interesting, but I just wanted to have your thought on like microfluidics and stuff like lung on the ships. Do you think it's the future of culture, or is it like crazy expensive and not as easy as LI for the future?
Yeah, I mean, I didn't actually, I had lung on a chip in my talk, Mathieu, but I just thought that the amount of people who have access to microfluidics and the ability to do that is pretty limited at the minute. It's very expensive. I think the future may be in lung on a chip, you know, I mean, The issue I always have, it comes back to that translational proteins. It's great to know the lung on a chip and know the DNA and the RNA and what changes, but it's actually doing the proteomics on that, you know, and I think it's really important that we don't get blinded by just the genetics of PCD, that there's more complexity in the post-translational mediation of the proteins compared to, I mean, the genetics are very important, obviously, but, you know, to understand the bigger picture of what's going on in a lung on a chip will be an excellent method for the future. I hope that answers your question. But it's expensive and not many people can do it.
Excellent. Thank you very much. Katie?
I've just seen another question pop in from Nuria. So excellent talk, Rob. How many passages maximum of proliferation would you recommend, or better to go directly to transwells when confluent?
Oh, right, yeah. No, that's a nice question, Nuria. I think the problem is that it depends what you want to do, really. You know, for us diagnosing PCD, the doctors need to know straight away. So, you know, putting them straight onto a transwell from the biopsy may be a better way than we're doing it, you know, because that gets the answer quicker. You have PCD or you don't have PCD. But if you want to kind of look at them over time, it might be worth, you know, doing at least one or two trips in EDTA passages in order to expand them out, so you've got more tissue to study, so you can apply your TEM and all the other immunohistochemistry for the cilia as well. So, I mean, it's a trade-off really, because obviously if the biopsy is small in the first place, if you then try and push it, you end up with that 20% failure rate, which we all see. So I think the answer is it depends what you want to do with it, but I wouldn't push them too far for PCD diagnosis. That is, for experiments, you can do all the different treatments, rho kinase, beta cells, you can BMI1 transform them, and you can study really good paired samples then, because they are identical, essentially, like a clonal cell line. So it's great to do that. I hope that answers your question.
Thanks, Rob. So like Matthew, I have lots of questions that I could ask, but I'll pick a few. Your TER values, what do you think causes your TER variability, and have you looked at macromolecular permeability?
Yeah, I mean, I did touch on it in the talk that I think what happens is certain patients, when you culture them, have more apical fluid than others. So I think that people have different ability to export from the basolateral compartment to the apical compartment. Now, whether that's because of more aquaporin production or more sodium potassium pump production or expression, I don't know, but it's certainly an interesting phenomenon, and it frustrates me a little bit, because the TER readings are so useful and so handy, but at the same time, within patients they're very, very reproducible, but across patients they're not. So that's where I get a little bit nervous when I have to review a paper where people have used it extensively, and I think, is that true? Is that a truism, or is that kind of just variability naturally in people's epithelial ability? The other thing that I've noticed is that the ones that ciliate and have a small, less mucus produced. So if you get someone who doesn't produce a lot of mucus and has very good ciliogenesis, the TER readings tend to be lower. So it's almost like if you get more mucus, you're going to get more resistance. So are you actually reading into those TER readings actually mucus production ability? I don't know. So you're right, it's important that we consider the macromolecular, what is going on when you take those readings exactly. And you need to both use the TER readings as a support mechanism to then look also at other things that may be going on in that cell type. I hope that's clear.
No, that's great. Thank you. You might have said it already. What temperature do you do high-speed video microscopy analysis at, and why?
Okay, so we always do, we have a heated stage, chamber slide, and we do the high-speed video readings at 37 degrees C. We always have. And it's important to make sure that you get the temperature correct. And we use a micro thermometer probe inside the chamber slide to make sure that the objective lenses, when we look at them, aren't a heat sink and suck the heat away, because that can affect the frequency. So it's also important to equilibrate the cells. So we leave them on the heated stage for a little while, maybe 5 or 10 minutes, just enough to heat up before doing any readings. The reason why do we do it? Well, we do it because that's the human body temperature, essentially. You look at other papers, and we've considered doing lower temperatures, room temperature and all that. And I can see the advantage, because you don't need all the extra kit. And also, arguably, is the nasopharynx a lower temperature than 37? Probably, you know, but we wanted to standardize our protocols, and we think that 37 degrees C is probably what the cell temperature is likely to be, because of the blood flow and everything else. So that's why we do it, because we've always done it.
That's a fair enough answer. I mean, we always do things for 7 minutes and 7 mils, just because that's how we've
If it works and you get consistency in your readings, that's the way to go.
We have a question from Lydia. Do you have any tips or experience for culturing cilia in other tissues, for example, female reproductive system?
Yeah, I've actually worked with the repro guys here a number of years ago now, and they were taking fallopian tubes out of ladies, and I was doing the primary cilia imaging from them, and they are very, very tricky to handle. Now, I know that there are teams in the world who have managed to grow fallopian cilia in culture, but I don't know whether the same system would work. You'd have to kind of do a literature search and email someone to ask them, because there may be something more specialized in there. The difference is obviously that, you know, these are exposed to air, whereas in the fallopian tube, they will be fluid filled probably. But it's certainly something that we need to look at. The same could be said for brain cilia, but no one's going to donate a brain brush in, I don't think.
Probably not. Thank you, Rob. And the next question is from Mathieu. He said he had the same question about tea, especially for the well-ciliated one with a resistance under 400 ohms, that we would usually consider infected or not looking good. Do you think it might be influenced by the number of cells you see in the TW?
Yeah, no, I mean, that surprised me, Matthew, because, you know, 400 ohms is like a failed culture, you know, but looking at it on the actual transwell, it was fully ciliated, and it was, you know, a thin cell layer. It was, instead of having the 2 cells growing, the basal cells, essentially what were there, are all differentiated into ciliated cells, and they were ciliated, and there was a seal. But yet the TIA reading was very, very low, and, you know, I was worried that it was a failed culture, but it wasn't. And it went on, and we kept culturing it and culturing it, and it was fine. So I suppose what I'm trying to show there is that you can get low readings and have healthy cultures, you know.
And I had the opposite. I had, like, you know, 1,000 ohms and no cilia.
Well, that's it. Yeah, that's even worse, isn't it? That's like scratch your head time.
Thanks, Rob. So we have another question from Robert. Hi, Rob. Great talk. You mentioned a review that is good looking at single cell identities. Could you say the author names again, please?
Yeah, Pascal Barbary they work at the, I think they work in a beautiful place. They're down in the south of France somewhere. And the lady who is an absolute up and coming star with these things is a girl called Laurie Emmanuelle Zaragoza. And they published a review article. Marie Legrande, she works in Paris. She does all of the genetics for the whole of France. Absolute legend in the field of PCD. And Hannah Mitchison, of course, Hannah. The three girls put together this really nice review, and that's worth looking at, worth reading. But Pascal Barbery's team, and also I think Laurie Emmanuelle now is a PI almost, so she's kind of taken the RNA-seq work and some of the stuff looking at differentiation pathways, and she's doing most of that work at the minute.
Thank you very much, Rob. I have a question. You've listed some problems here that you have encountered during developing and improving the cell cultures. What step would you still like to improve, or any step you'd like to introduce to mimic more the in vivo environment?
Yeah, I mean, you're never going to get an in vivo environment looking at these kind of cultures. They're never ever going to be anything like the in vivo scenario. So Matthieu touched on it. There's a microfluidic way where you can actually recapitulate the lung on a very, very small chip by adding smooth muscle, fibroblasts, and then you can rebuild essentially the lung. So that would be the way forward. It's an incredibly technically difficult thing to do. Only a few groups in the world have actually managed to do it. And right now the data isn't really very useful at the moment, but it will be probably in the future. So I'd like to move towards trying to regrow the lumen. We don't know anything about branching of the lumen. We don't know about the there's suggestions that the nasopharynx is different immunologically to the lower airways, and that the lower airways, the larger airways, are different to the bronchioles and the very, very small airways. So we need to really try to consider how we're going to study these different parts of the lung to understand disease and disease progression better. And I know that Amelia is doing a lot of this, looking at bronchiectasis. She's looking at mucus plugging and how that causes death of the peripheral airway, alveoli and things like that. So, you know, all these techniques are great, but you can only answer certain questions with them. So yeah, any improvements, just watch the literature. There's always someone somewhere doing something very, very amazing.
Thanks, Rob. Katie, I think you have some more questions.
I've got one about the read-through therapy. So can you speculate as to why ciliogenesis isn't happening? Is it, you know, proteins aren't being targeted? Why is the protein not functioning?
Yeah, well, we touched on it in the paper. It's a really tricky one, MCIDus. It's multicillin is the protein, so MCIDus is the gene, multicillin is the protein. It's quite a large protein, and I think what's probably happening is the atellarin is actually helping to produce MCIDus, but I think that the stop codon, you know, may actually introduce read-through errors in other things, in other transcription machinery later down the line. There may be post-translational modifications that none of this read-through therapy can help with. And I believe that what we're seeing is a partial reversal of the MCIDus mutation, where we're getting enough signal through for the cells to say, right, we can make multiple cell bodies, but not enough to drive ciliogenesis. I'm guessing. If hand on heart, I'm guessing there, Katie. But
Great. Does anybody have any more questions? No? So with that, we'll wrap up and say a massive thank you to Rob for an excellent presentation.